Okay, so let's take one last look at the lactase region in us humans with Gillespie's model in mind, and then move on to consider some of the implications and predictions from the model. Here's a map of the gene region showing lactase here on the right, with the site where the European lactase persistence mutation occurs, which is in the middle of an intron of the neighboring gene, MCM6. Here's the exact spot. It is inside the binding site of a known transcription factor footprint. So it's very plausible that that mutation changes the binding of the transcription factor in a way that switches on expression of lactase even after the time of weaning in a mammal like us that normally would shut it off after weaning. And here's that figure of Allen's again, showing the just a sampling of the much larger number of chromosomes that are all identically the same in the neighborhood of the new derived mutation, this A, in the transcription factor footprint, which pretty obviously was what triggered the very rapid rise of this particular chromosome pattern to very high frequency in Northern Europeans, leaving behind a relatively small number of older, highly recombined and mutated chromosomes that reveal their age and their diversity by that pattern that we see in the bottom. This just describes what we've seen before. I'll just emphasize again that these 101 variable sites are embedded in and span over a large region of roughly 140,000 base pairs. So the vast majority of this region is completely identical in all the chromosomes. We're just focusing here on the sites that are variable and that let us see this pattern of improbably large number of chromosomes that have not diverged at all in this region, or only very slightly. For example, this T here and a few other very low frequency new changes. There is some verbal sense, anyway, in which you can think of, or we are tempted to think, of these consensus chromosomes as younger. I would just invite you to think about whether that really makes sense. What is it that's younger about them? I don't think it's their mutations. It's not even really the combination of mutations, but it is the fact that they've all exploded as a young family, as it were, derived from an ancestor that looked just like themselves. A more enlightening exercise than arguing about the meaning of the word young might be to try to identify a few of the recombination events that can easily be found in among these quote-unquote older, more variable chromosomes, where you can actually see where crossovers had happened in the past. This figure I made from the HapMap data for the 90 Utahns in the HapMap dataset to show where these individuals have tracks of long-range homozygosity. So these thick red lines, each vertical line is one individual's diploid genotype, and the thick red patches show where that individual doesn't show any heterozygosity for an improbably long distance. And so that view clearly indicates this region, which is where the newly swept up identical consensus chromosomes lie, which isn't centered on the lactase mutation, which is actually here toward one side of it, again reflecting probably the early history of random recombination events that happened here, for example, and here, and here, and here, and so on. Okay, it is the case that in this zone of bizarre identity, the overall heterozygosity, including all the chromosomes, is drafted down. It's lower on average than in regions of chromosome 2 far away, but to my eye, it's not very dramatically so. Right? This valley of reduced heterozygosity does call itself to our attention once we know that that's the place where the consensus chromosomes occur. But if you didn't see the right-hand half of this figure, you probably wouldn't think anything was particularly special about that. So that raises the question, why is the effect so irregular and modest? You could play around with a simple little Python simulation that Gillespie puts in the book here at the end of chapter 4. I warmly invite you to work this up. For Python 3, you'll have to fix this print statement and possibly a few other things. It's just a deterministic simulation that lets you see the ending heterozygosity at the B locus for different initial conditions, and the way to make it more enlightening than it is in this version would be to try a bunch of different values of the selective advantage of the sweeping new mutation, as well as the recombination rate between the A and B loci, and also to consider, which Gillespie does not do, initial frequencies of the B locus alleles other than 0.5. He starts them all at a frequency of 0.5, which seems needlessly boring. And I think if you play around varying all three of those initial conditions, you'll get a feeling for how variable the outcome of draft may be, so that it does seem consistent with the pattern I just showed you around lactase. Okay, so now what about the general predictions of the model? They are obviously, first and foremost, that there should be less variation on average where sweeps, selective sweeps, are more frequent, and where their recombination rates are lower, because then the zone that is swept out will be wider, and so the total amount of genomic variation produced will be greater just because the sweeping is over a wider path on the chromosome. Many years ago, as a graduate student, David Bagan and his advisor Chippa Quadro did this beautiful study of synonymous heterozygosity on the X chromosome of Drosophila melanogaster, exploiting the fact that a lot of genetic work had established variation along the chromosome in the rate of recombination, which they call the coefficient of exchange in this graph. On the left-hand side are low values, meaning very low recombination probabilities. On the right-hand end, some rather high recombination rates. And what they discovered, a totally novel result at the time, was that the level of synonymous site heterozygosity in coding exons was significantly lower in the regions of low recombination, that is, in genes that were parked in the middle of regions of low recombination, than in regions of high recombination. They considered an alternative hypothesis, probably suggested by a referee of the paper. What if the recombination itself was mutagenic? Maybe that's why zones of higher recombination have more heterozygosity. Maybe the mutation rate is higher. And obviously they thought about that long and hard, came up with a lovely way to show that it wasn't plausibly true, So my challenge to you is reinvent their test. Hint, it involves some comparisons between closely related species. Maybe you can figure it out with that hint. If not, or in any case, you should read the paper, which is short and sweet and well worth your time. You can easily find it. I think it's in Nature. So what determines the frequency of adaptive sweeps, which is really the key parameter driving this whole process? Gillespie gives it a symbol, lowercase Greek rho, so that looks like P, but actually it's rho, lowercase. Well, of course, the ecological engine of adaptive sweeps is the rate of environmental change, which we think of as inducing a species to change its ways somehow, but also given a certain rate of environmental change, the rate of sweeps is going to depend also on the rate at which adaptive mutations arise as mutations within the population. And Gillespie thinks that through in chapter 4 and comes up with this enigmatic and hard to understand graph that Alan and I have scratched our heads over, and I'm going to try to help you find some sense in it by walking you through it. It shows on the y-axis the effective population size, by which he means NE itself, the effective population size, and also its correlate that we can see, which is pi, the nucleotide level heterozygosity, especially at neutral or silent sites. And he considers, he means to contrast here, two alternative stories or models for the way in which adaptive sweeps occur. The simplest model is one of a constant rho, that is to say, adaptive sweeps are purely, their rate is limited by the rate of environmental change, not by the rate at which adaptive mutations occur. And in the book, I won't drag you through it, he gives an argument in the chapter for this little model that predicts NE as a function of the actual population size and rho, this rate at which adaptive sweeps happen, which is a small number. Sweeps per generation are its natural units. And then this thing, the expectation of the squared final frequency of the lucky hitchhiking gene copy. If it always went to fixation, y squared would be 1, because y would be 1, its final frequency would be 1, but of course it doesn't always make it all the way to fixation, as we saw, and so you have to, it turns out that a number which is the expectation of the squared final frequency wants to be part of the numerator below. Okay, so NE is the actual population size devalued by this denominator, which is greater than 1, and it goes up with n and rho and this expectation of y squared thing. All right, so if rho is just a constant, then it turns out the effective population size will increase rapidly at first with the population size, but then turn over and saturate at an asymptote that is not reflective of the population size at all. So that's kind of consistent with that original Bazin et al. Figure for the mitochondrial variation anyway. Remember, the nuclear variation did seem, at least on average, to sort of go up with population size. Maybe that means that we're in this part of this curve for the nuclear genes, but because of something to do with mutation rate or mitochondrial biology, we're more in this part of the curve for mitochondria. That's one of the mysteries of this subject, still remaining to be worked out, I would say. Okay, but the other interesting and to me very novel idea that Gillespie presents to us is, well, what if the rate of adaptive sweeps was actually mutation limited? Then it might depend at the end of the day on the number of new mutations that happen, in particular at loci capable of mutating to adaptive substitutions, and so he invents a new mutation rate, little v, to represent that. S is the usual selection coefficient, so a selective advantage, n is the population size. Okay, so he considers then this alternative style of model in which it's not environmental change that limits the rate of adaptive sweeps, but instead it's the creation of the adaptive mutations themselves that will be positively related to the population size, because in a big population, other things being equal, more mutations means more adaptive mutations, and so on. And so then it turns out with this rho term being like 4n times something small. In here, we have an n squared term, and so after an initial sharp rise with population size, ne and pi turn over and start crashing as the amount of adaptive sweeping goes through the roof with increasingly large population size, right? And so paradoxically, under this model, at least over certain ranges of its parameters, you could have a truly insane inverse relationship between population size and nucleotide diversity. And it is conceivable that that is part of what explains the story I presented in the very beginning about the bizarrely low levels of mitochondrial neutral diversity that we see in the Antarctic krill, the species with arguably the largest population size in the world, at least of a macroscopic animal. But if most species environments change at the same rate, because nature is nature, then they might turn out to have strangely similar values of the effective population size and pi, the way we estimate effective population size, regardless of their true n. To repeat that point again, which is also what we see over and over again in the data. The Bazant paper shows one data set that shows it one way. Here's an old compilation of observed levels of pi, nucleotide level pi, from early in nuclear gene DNA level population genetics. More modern data sets would show the same thing, which is that while pi does vary overall by maybe a factor of 100, most of the variation is actually concentrated over a range of about 1 order of magnitude only, from something like a few tenths of a percent, that would be us, and well, we're even slightly below that, but we're weird, up to maybe 2%. But in pretty much all cases, the observed levels of nucleotide polymorphism are lower than we would expect given the neutral theory. That is, they're quite a bit lower than reasonable estimates of 4n mu. Okay, so our summary, draft is the effect of a selective sweep at one locus on the variation at nearby, that is, genetically linked loci. On average, it clearly will reduce variation at those linked loci, and in this way, it reduces the apparent population size, and as Gillespie's argument, I think, convinces all of us, that really is the effective population size for many purposes. So effective n's are smaller than you think. Gillespie calls it a new stochastic force in evolution arising from the stochasticity of the initial conditions, which is different from the source of other stochasticity like genetic drift. That is, it's the stochasticity of when and where in the genome a favored mutation first occurs. I myself don't think it's really a new force. I think Alan agrees with me in this. It's more of an effect, as I implied at the outset, but whether you think it is an effect or a fundamental force or process, it's certainly important. And apparently we don't yet understand the whole story, which is to say how it is that many sites in genomes get into states where they are eligible to have a new adaptive mutation happen at them. It's when you stop to think about it, it's not so obvious that that should happen very often. And in any case, we don't have a good theory for what sets the rate at which adaptive substitutions can and do happen. Okay, so that's the end. See you on another subject soon.